Chair. It's Jeff Ross. I'm CEO of Miromatrix Medical. Madam Chair, members of the committee, it's a delight to be here and just kind of share some of the stories of a biotechnology life science company that was founded here at the University of Minnesota, and that we continue to move forward with a very grand mission. And our mission is to solve the organ transplant shortage through developing transplantable organs. What I want to do today is just kind of give you an overview of who we are, talk through some of the stuff, but some of the challenges that we've done along the way to give you a little bit of a sense from the time of startup all the way through clinical trials. What does that really look like for a product of this nature that could have a major impact on the healthcare system, but certainly the economy as well? So if we look at this, Miromatrix is developing a revolutionary solution to solve the organ transplant crisis and eliminate the waiting list. We essentially are changing medicine. Our approach is based on technology that was developed at the University of Minnesota, and this is called perfusion decellularization. And that's the ability to take a whole existing organ, and we actually cannulate it, meaning we hook a tube up to it, and we perfuse a mild detergent through it. What that allows us to do is wash out all the cells. Kind of describe it like remodeling your house. If you think of drywall as the cells inside of an organ, we essentially remove that. What that leaves you with is the perfect scaffold, and I'll show you some images of that in a second. We're then able to place that into a bioreactor, which you see in that middle column there, and then perfuse human cells back into it and essentially reanimate that organ to be able to create transplantable organs. We've done a ton of de-risking. We actually took that first base material, which is a whole porcine liver, perfusion decellularized that, and we created medical devices with that. We launched two products, one called Miromesh, one called Miroderm. Got those through the regulatory process, manufactured those in Minnesota, and those are being transplanted into humans today. That's the base material that we're going to build our human organs off of. We have a lot of IP. I'll show a slide in the end. A lot of cost goes into developing an IP strategy, but more importantly, getting patents and being able to protect those patents as well. If we look at our programs, what are we trying to develop? Our lead program is a transplantable liver. Large market opportunity, just within the U.S., is estimated over $10 billion. And then also to go after kidney and create transplantable kidneys. I'll talk about that in a second. To date, as an entity, we've raised $47 million. Our last round was a $16 million round that we also brought in a large strategic. Currently out fundraising right now for an additional $40 million for the Series C, which will bring us all the way through enrolling 5 patients into our phase 1 clinical study for a transplantable liver. Those are our plans. We're targeting that in 2022. Just to give you a sense of, if we're looking at something this grand, how much capital does it really take to be able to bring a product like this to market? Knowing that within medical devices, there's a large continuum, right? Some need relatively small amounts, some need larger amounts. So just to walk in a little bit, what are we trying to solve? If we look at this is a nice little chart here of just showing the number of patients today who are waiting for an organ. In America today, there's 115,000 patients. Each year, 80,000 patients do not receive an organ. So if we look at kidney alone, there's 95,000 patients who are currently on the list waiting for a transplantable organ. There's about 21,000 kidney transplants performed in 2018. The average wait time is 3 to 5 years. More importantly, I think the stat that really talks to itself is that ability that each day 13 patients die while waiting for a kidney. Another 8 patients are actually usually brought off the list because they're no longer healthy enough to qualify for the surgery to actually get a transplant. Transplant outcomes are far superior to dialysis. Similar with liver, the 17,000 patients are currently waiting on the transplant waiting list for a liver. There's no alternatives to if you're an end-stage liver failure, and there's about 40,000 deaths associated with that. So there's no drugs, there's no dialysis, there's no devices. So unfortunately, either you receive a transplantable liver or you really don't have any options. So if we dive into this a little bit, I won't spend a lot of time on this. I mentioned livers are lead for that reason, that there's really no alternative therapies that exist out there for liver therapy. And what we're planning to do is develop a transplantable liver to be able to solve that need. Where are we to date? I'll talk a little bit about the timelines that were associated with that as well. But we've already met with the FDA. We had our Interact meeting, so that's the pre-IND. That's essentially where we talk about all the data that's going to be required for us to get into human clinical studies. We believe that we're the only company that's been granted an Interact by the FDA at this point, just demonstrating how far ahead we are inside of that process. And again, we're planning our phase 1 clinical studies in 2022, and then targeting orphan status just because there's no alternative therapies for it in 2024. Kidney even gets more amazing by the number of patients and the ability to be able to affect these patients. If we look at today, there's about 500,000 patients in the U.S. who are on dialysis, with 100,000 patients annually added on dialysis as well. But I think what a lot of people don't know is the outcomes are incredibly poor with dialysis today. So if we look at it, 20% of patients die within 1 year, 50% of patients die within 5 years. Today, those outcomes are worse than many of the cancers that exist in terms of diagnosis. Renal care really hasn't kept up a pace innovation-wise compared to other sectors inside of our healthcare system, which is another thing that is helpful in the fact that an executive order earlier or last year on advancing American kidney health provides clear momentum. And they're really calling for, how do we invest in this sector? How do we create a bioartificial or an artificial kidney to really give these patients a new option? As part of that, they spun out an entity called Kidney X. It's a private-public partnership, and we were one of 15 teams that won a phase 1 award. Why that's important, there was 165 teams that applied to win that award. And really the focus is on how do we create new renal therapies. So that's really what we're going after and why we're trying to create this. So how are we going to do this? I talked a little bit in the beginning just to give you a sense on what our technology looks like. And that's the ability to be able to take an existing organ. We take them out of pigs because it's a byproduct of our food process today. There's an endless supply, and we can be able to create that pristine matrix. We take that, use our process to decellularize that. That allows us that first product where we then have that perfect scaffold. We then take organs that were unsuitable for transplant. So for each organ that is placed into a patient, there's multiple organs that don't make it, whether it's from cuts, tears, nicks, trauma. We're able to source those. Now we have good, viable cells. We're able to isolate those cells out and place them onto that scaffold to then be able to recreate that organ to essentially increase the supply of transplantable organ. That's our phase 1. Phase 2 product, I won't talk much about this, but that's the long-term potential, and that's the ability to be able to engineer organs from stem cells. So whether they're patient-derived or allogeneic stem cells, but that ability then to be able to create organs that no longer need immunosuppression, kind of the holy grail. But that's further inside of the future associated with that. So at a high level, that's kind of what we're doing. Grab an existing organ, remove the cells, place new human cells on it to be able to create that long-term solution. So again, at a high level, what's our technology? There's a pig liver there. You can see we cannulate that. We perfuse a mild detergent through it. By 24 hours, it's completely white. It's essentially called a ghost organ at that point. In this video right here, I'm just going to demonstrate that as part of this process, if we reperfuse this with a red solution, you're going to be able to see that all the vasculature is intact still. So we don't have that ability of how do you recreate that substrate and have all that intact vasculature. Again, all that architecture is there. So we now just need to recellularize it to be able to create that end solution. So if we look at this and we kind of parse this out, how do we get all the way to from where we are, inception, all the way through? We created 4 buckets that we had to demonstrate. First, prove that perfusion decellularization is effective. Then how do you recellularize it? Can we actually put cells back onto it? And can they engraft in the appropriate geometry? Third, can we then implant that into preclinical studies, demonstrate functionality associated with them? And then that allows us to work with the FDA and then move into human clinical studies, demonstrate the efficacy there, and now you have final approval. Those are kind of the big buckets as we look through the process. So where are we? We've demonstrated perfusion decellularization is effective. We created commercial product. That's our Miromesh, Miroderm. Created a manufacturing process associated with that. Completed multiple clinical studies. So now we are demonstrating that that base material is incredibly effective. It's non-immunogenic. We're able to now build the substrate off of that. So the next step is, how do you now revascularize that? So how do you add cells back in and demonstrate that you can recellularize it? And I won't go into a lot of the data there, but we're able to source cells, grow those up, seed them back inside of the grafts. These are just a couple of images here, just demonstrating how this works. So this is a revascularized liver graft that's going back into a pig inside of this model. And what you're going to see is what this looks real time as we take those clamps off of it. This isn't overly bloody, so this should be fine. But what you can see, right, is now we're able to actually create a substrate. We're able to transplant that back in. When we take off that clamp, you can see that, that's essentially what a native liver looks like at that point. We then take off the other clamp. Now we've actually anastomosis back into the host, and we have the circulatory system going through it. So now we've demonstrated the matrix is okay. Now we've been able to revascularize this, and we can do this with a kidney as well. This is an angiogram after 10 days post-implantation, just demonstrating that we have that patent vasculature. So as I mentioned, we're able to take the matrix, we're able to add cells back onto it from the vascular standpoint, be able to transplant that back in. Now all of our focus is really on the ability of demonstrating in vivo functionality, of now adding the specific organ-specific cells to move to that next level. A lot of data on this slide, I won't go through it, but to demonstrate that, yes, we can now take liver cells, seed those back on. We can get liver function back out inside of our bioreactors. We also now are surgically implanting these and looking at functionality of those as well. So long-winded way of just saying things are going well. We continue to move in the right direction. And if we look at timelines from where we are today to where we want to be in terms of regulatory and risk, on the liver, we've really moved through the research phases. This gives you a sense of, how you need to move through from the standpoint of de-risking the technology, proving that your value proposition is effective, and then moving into the development phase, which we are right now in terms of rescue of preclinical animals to be able to work with the FDA to be able to support IND studies, do the IND submission, which then would allow us to get into phase 1 human clinical studies. Now, for a product like this is a full BLA. We would have to go through all the phases of clinical studies to get final regulatory approval from the FDA. In parallel with that, a lot of conversations go around reimbursement. What does reimbursement look like as well? To move all of those are working pieces that we continue to work through. If we look at the timeline associated with the kidney, very similar in terms of research phase, where we're at today, development phase, demonstrating that we can recellularize that whole kidney, place it back into a large animal model, and start to be able to bring back that renal function, which is that really big next step as we continue to target how do we create life-saving technology for dialysis. Won't spend a lot of time on this. Senior management, including myself, that was introduced, but board of directors as well, multiple individuals across the state, many of them current or ex-CEOs as well in the medical community. Just real quick on financing as we talk through this, just to give you a sense of what this looks like in the phases. So we were spun out of the University of Minnesota in 2009. In 2010, we initiated our first seed round that allowed us to create some of those early accomplishments. We then went into a Series A. Again, being able to then start to look at what that core patent looked like, protect the IP to be able to build a company around that. Then had a Series B, which allowed us to launch those first products and de-risk it, and then a B2 that allowed us to start getting some of that preclinical study that I just demonstrated to you. C, our next funding round, then sets us up to finish out those efficacy studies in our preclinical models and then move into human clinical studies. Have a very strong IP portfolio, so all 3 of our parent patents have now issued. We have 7 issued patents in the U.S. and covered in all OUS markets as well. This is just, I just threw this in here as a comparable. So what would this mean as we continue to go forward if we're successful in bringing the product all the way to the market? It brings tremendous value to patients, but also, I think, to the ecosystem and what we're trying to achieve here. So that's Miromatrix.
Thank you very much, members. For each of our guests today, we're going to take a little bit of time for questions. So my first question, I saw on the slide, phase 1. Could you walk us through just what the phases are for product development? And this isn't really a product. I don't know how to describe this, but FDA interaction.
Sure, Madam Chair. So if we look at, there's different classifications of products, right, depending on the risk profile, if they're class 1, class 2, class 3 with the FDA. Some of those can go through a regulatory process called a 510K, where essentially if there's an equivalent product that already exists on the market, you then can file and make the argument that your product is similar to that. What that allows you to do is void clinical studies and get that product approved, so then you're able to sell that product. For class 3 and products like this, there's some exemptions in class 3, but But for biologic-based products like what we're developing, pharmaceutical products as well, you need to move through that full pre-market approval, PMA, to be able to then demonstrate safety and efficacy for the FDA to give you final approval of those products. So that's usually phase 1 is usually a safety study. So that'll be a small study in pharmaceuticals. That's your dosing study where you'll apply different doses and look for safety. Phase 2, you start to get into some level of efficacy to be able to look at, does your product work? And then in phase 3 is usually your pivotal study. So that's where you actually power it to demonstrate that, yes, it's having the intended effect that you have, and that's where you'll get your value props. With biologics like what we're developing and what you're starting to see with genetic gene therapy that's starting to come out, now you're starting to see those first approvals in some of the cell therapies called CAR-T that are going after some of the non-tumor cancers. What you're seeing is much shorter phase 1 and phase 2 and smaller pivotal, because it's a lot easier to demonstrate efficacy. What I mean by that is that if a patient doesn't have a gene, in your phase 1, you're already proving efficacy because you can quickly test, do you have that or not? Similar with ours, as we look at our product going forward, when we do liver transplants and we do kidney transplants, even during the safety study, you're gonna know, do you have function or not? So that allows that to shorten that window somewhat.
Excellent. Thank you. Senator Jensen, did I see you?
Thank you, Madam Chair, and thank you, sir, for your presentation. I was just curious about when we take the scaffolding, if you will, from the pig and wash it and then put the human cells back in, and you get the product and to the end point where it's ready to be transplanted. How does that compare to some of the research where we're actually growing the human organ in a pig? We take and that how, in terms of a total price before you go, and then also outcomes in regards to successful transplantation without rejection. Thank you.
Right. So, Madam Chair, Senator Jensen, great question. So for the first one, this is a huge area of development, right? True need. I mean, there's tons of organs that need to be developed. So there are a couple of technologies. Ours is one approach, the ability to take a matrix, add human cells onto it. Our approach really looks at, if we look at from the immunogenicity side, since we're recellurizing with human cells, we anticipate that we know what that immunosuppression is going to need to be able to do, because it should be similar to cadaveric organs or living donor organs today. There's another approach, and that is, well, can we just genetically modify the pig to be able to take that animal, to take that organ directly out of the animal and place it into humans? So that's called xenotransplantation, and there are multiple companies working on that approach as well. Probably been working on it for the last 30 years. Originally, they got hung up with a couple of proteins called alpha-gal and some other ones that we have a ton of antibodies to. So the problem is you can't take an animal organ, put it in a human, because we immediately reject it. So they are using current technologies such as CRISPR and gene modifications to be able to modify that genome to see, can we get it to the point that you can transplant it into a human? And, you know, I think the verdict is still out. I think they're up to 15 or so genes that they're currently knocking out to be able to try to get there. But the open verdict, I think there, the unknown is even once you get past the immunogenicity, A, can you get past it? But B, from a functionality standpoint, will it function the same, right? Body temperature is a little bit different. Some of the receptors are a little bit different. And I think that's still the unknown on that. Where on our approach, as we talked about, right, we've already de-risked the porcine scaffold once it's decellularized, because our current products, to de-risk and take that immunogenicity off the table, essentially, and then add human cells onto it.
Thank you. Thank you very much for your presentation today. Truly impressed, and given how much we spend on dialysis, it'll be interesting to see how your approval process goes, and then how we get this into our market. So I thank you for your time today. Oh, Senator Rosen.
I'm so sorry, Madam Chair. Thank you. Mr. Ross, is this a very competitive market?